Today we're going to talk about cell membranes. And this is kind of an important topic, right? We've been talking about membranes before in the context of eukaryotic and prokaryotic cells and saying, okay, prokaryotic cells lack membrane-bound organelles, while eukaryotic cells have membrane-bound organelles. And if we think about the structure of a cell, it has a plasma membrane or an outer membrane that surrounds it. So membranes are kind of crucial to our understanding um, of the structure of cells and the fact that these membranes are creating little areas. So organelles inside of cells have the membrane that surrounds them so that certain chemical processes can take place within that structure. So we're kind of segregating all the enzymes and the molecules that are needed for a particular function to happen within that organelle. And we're creating that organelle by creating a barrier or putting a wall around it in the form of this membrane. So when we make these membrane-bound organelles, it's simply referring to the fact that there's a membrane around them. And we have a membrane around the cells. Same thing. We want to be able to regulate what goes in and out of the cell. We don't want just any old thing to cross into the cell, and we don't want to lose things from the cells. So the membrane is going to help to act as a barrier that will regulate what is transported in and out of the cell itself. So we need to look at the construction of a cell membrane. We know that phospholipids and cholesterol are important in cell membranes, but we haven't really talked about how they all go together. So membranes are what we call a bilayer of phospholipids. So a bilayer meaning there are two layers. So here's our phospholipid molecule. We've talked about it before. We've talked about the egg on legs model where we've got this kind of egg-shaped head region to it and two tails that are consisting of fatty acids that are coming down off of it. And we have cholesterol here, and we've got these big kind of chunky purpley structures here. These are all different types of proteins that are embedded in this membrane. All right, so all this is a membrane with the phospholipids and the proteins collectively together. All of it is considered to be a membrane. Now, we said it's a phospholipid bilayer, so two layers of phospholipids are the main component of our membrane. If you remember, our phospholipid had this head region that was hydrophilic, and that was because it had a phosphate group where it had oxygen attached to the phosphates, and this gave charges. And anytime we said you had a charge, it would attract water. And the two tails were comprised of nonpolar covalent bonds. These are hydrophobic. So if we think about it, typically a cell membrane is separating two aqueous solutions, right? So the liquid that's kind of on the outside of a cell could be filled with water, and the contents within the cell are usually water-based as well. So we have this phospholipid molecule that has this dual nature of being hydrophilic and hydrophobic. So the hydrophobic tails of the phospholipid are not going to want to be anywhere near the water, but we do have at least one end of it that likes the water, and that's that phosphate group or the head region of the phospholipid molecule. So we cannot have just one layer of phospholipids because then the tails would be in contact with water, but we need two layers. And you'll notice the second layer is inverted, or it's kind of upside down compared to the top layer. And that's so that these fatty acid tails that are hydrophobic are tucked away from any water source that might be in or out of the cell. So having a phospholipid bilayer is important because of the nature of the phospholipid molecule and the fact that it has hydrophobic regions that are not going to want to be in contact with water at all. Now, a membrane has to be fluid in order to function properly. So that probably sounds kind of weird. We probably like to think of membranes as more of a solid, but they're actually a fluid. These little phospholipid molecules can flow past each other within their layers. Now, we don't want it to be too fluid though. We don't want our membranes to be very runny at all. But we need it so that we can squeeze in between these little phospholipids and move molecules across our membranes. So we want it to be a barrier, but not a solid barrier. Okay, so they have to be fluid to work properly. Now, we notice the difference between this image on the left and the image on the right. The image on the right, we've got a lot of phospholipids that are packed in nice and close to each other because their fatty acid tails are very straight. On the left, we have fatty acid tails that are not so straight. They have a kink in them, a little bend. You can see they're bending at different angles where they occur. So if you remember when we were talking about saturated and unsaturated fats have double bonds between their carbon atoms, and that will put that little kink in that chain. Saturated fats are saturated with carbon and hydrogens, or really the hydrogens, and therefore that makes a nice straight chain. So these phospholipids would have all saturated fats making up their fatty acid tails. Over here we have some unsaturated fats, the double bonds that are putting that kink in there. And notice what happens. Because there's a kink, these phospholipids cannot pack as tightly or as close together. All right, so because of that, having unsaturated fats keeps the membrane from solidifying. If the membrane becomes a solid and solidifies, then it's very hard for molecules to pass across it. All right, so this one on the right would be more likely to solidify because they don't have the unsaturated fats. Down below here, we have more phospholipids, but now we have a different molecule with ring-like structure, so this is cholesterol. And cholesterol is in between our phospholipids. And we see it kind of a little further down. We see another cholesterol molecule, and it's on both sides of our bilayer. So cholesterol can actually keep our membrane from being too fluid, from keeping these phospholipid molecules from flowing too far. So it's almost like we're placing a little fence or and basically saying, okay, these phospholipids, these 6 phospholipids can move between this cholesterol molecule and this cholesterol molecule, and that's as far as they can flow. All right, we don't want them to keep on flowing all along. We want them to keep it within this little area here. Okay, so our unsaturated fats can keep our membranes from becoming solids, and our cholesterol can keep our membrane from being too fluid, just the opposite of that. Now, besides the phospholipids, we mentioned that there are proteins embedded in the membranes, and there are a wide variety of different types of proteins. And each of these different types of proteins performs slightly different functions. So we're going to talk about these proteins very generically and talk more about kind of what is their purpose, what is their function, and not necessarily give names to them. So there are what we call receptor proteins, and that's their job, is to receive chemical messengers. So they are embedded in the membrane themselves, and they typically have some type of binding site where a messenger, a chemical message, can be received by that protein. So once again, we got a little egg on legs bilayer that we're seeing. The big purple circle is our protein, our receptor protein that's embedded in our membrane, and in orange is our chemical messenger. So that little kind of cutout pie shape wedge there is our area of our protein where our chemical messenger is going to come in and bind. So the shape of that binding site typically has to match the shape of the chemical messenger, and this prevents it from receiving other messages. So once it receives that message, it can cause a change in the protein that is usually then relayed to the inside of the cell to say, hey, this is what's going on outside. We need to make some changes accordingly. So it could be that this protein here is receiving a message that they are low on human growth hormone, and therefore it's time for the cell to start producing more human growth hormone. Okay? Now, our membranes are what we call selectively permeable. They're very selective as far as what can cross over the membrane on their own. So some molecules can pass on their own, but others tend to be excluded, and that has to do with the construction of the membrane. So if you remember, we said there's a hydrophobic core in the middle of our bilayer due to those fatty acid tails of our phospholipid molecules. So anything that is hydrophilic is going to have a hard time passing through that hydrophobic core. So anything such as an ion, which is a charged molecule, it could have a positive or a negative charge, or even large polar molecules. They have partial positive and partial negative charges. They'll have a hard time getting through the hydrophobic core and typically will need the assistance of what we call a transport protein. So other molecules that are hydrophobic, or even some very small polar molecules such as water, can pass through the phospholipid bilayer on their own. So water can pass through. It's a very small molecule, even though it's polar, but it passes through very slowly. Okay, so it is possible, as long as it's small, it can pass through. If we give it a transport protein to aid in assistance, though, water molecules will pass through much more quickly through that transport protein than if they were just traveling on their own through that hydrophobic core.